Hello students, welcome to EPG Pathshala. I am Dr. Renu Tyagi from Sardar Patel Institute, Ahmedabad. Today we will learn about the module Biochemical Parameters under the paper Physiology and Sports Anthropology. Now let's have a look on the broad objectives of this module. After going through this module, the learner would be able to know the structure of the hemoglobin and the function of hemoglobin. We will be studying various laboratory methods for the estimation of hemoglobin. We will study advantages and disadvantages of each method. And then we will study the basic statistical concept in the quantitative hemoglobin data presentation. The hemoglobin, the pigment of the red blood cells, is responsible for the transportation of oxygen in the blood. This is of great interest for the anthropologist as well as for the clinicians. There are primarily two major attributes of this red cell constituent that has been managed to draw much attention of the scientific community. That is the polymorphism of the hemoglobin variants and the quantification of hemoglobin in blood for assessing anemic condition. Till date, 479 genetically controlled hemoglobin variants, that is the variation in normal hemoglobin molecules, have been identified. Though majority of them are rare, few such as the sickle cell, that is HbS, hemoglobin D, HbD, and hemoglobin E, HbE, and some other are polymorphic in many population, thus provides an ideal tool to study the population variability and affinity. However, the scope of the present module is limited to hemoglobin quantification. Thus, an attempt has been made to discuss the different methods of hemoglobin estimation in the field conditions, especially in the situation where the resources are limited. This module elaborates the most common methods of hemoglobin estimation, and it also discusses upon the other available methods. Hemoglobin is the major constituent of the red cell cytoplasm, accounting for about 90% of the dry weight of the mature red cell. The hemoglobin protein macromolecule is a tetramer constituting of two pairs of similar polypeptide chains called globin chains. Two of them are alpha chains and two are beta chains. To each of the four chains is attached the heme, which is a complex of iron in ferrous form. The major 96% type of hemoglobin present in adults is called hemoglobin A, and it has two alpha globin chains and two beta globin chains. The genes that code for the formation of a alpha globin chain is located on the chromosome 16, and that which codes for the formation of beta globin chains is on chromosome 11. In adults, a minor amount of another type of hemoglobin, that is hemoglobin A2, is also present and constitutes less than 3.5%. During embryonic and fetal life, other different type of hemoglobin predominates. These include the GOVR1 present in early embryonic life, GOVR2, and hemoglobin Portland. After the 8th week of the development, embryonic hemoglobin is replaced by the fetal hemoglobin. This remains the predominant hemoglobin until after birth and constitutes the 50 to 90% of total hemoglobin. After birth, its concentration decreases to less than 2% by 30 weeks of age. Hemoglobin A is then the predominant hemoglobin. Now let's have a look on the functions of hemoglobin. Heme has the ability to bind the oxygen reversibly and carry it to the tissues. It also facilitates the exchange of carbon dioxide between the lungs and the tissues. Thus, hemoglobin functions as the primary medium of exchange of oxygen and carbon dioxide. Significance of hemoglobin estimation is studied by screening test for detecting the anemia. This is a frequently identified abnormality in the population. Anemia is not a diagnosis by itself, and if detected, its underlying cause must be ascertained. Hence, accurate hemoglobin estimation is essential so that further tests can be done to ascertain its cause and the patient can be treated accordingly. The methods of estimation of hemoglobin. The various methods are available for estimation of hemoglobin in laboratory and the field situation. Many of them are now obsolete with availability of newer and more accurate test protocols. In this module, we have discussed the 5 protocols in details. These methods are suitable to carry out at resource limited setting and in the field situation. Sali's or the acid hematin method, Cyanmethemoglobin method. Number 3, hemoglobin estimation through portable hemoglobinometer. Number 4, hemoglobin color scale method, that is WHO color scale. And number 5 is the copper sulfate method. The Sali's acid hematin method is the most commonly used method, and it's a most trusted test for the hemoglobin estimation in resource poor setting and the field situation. This test requires dilution of the blood and the visual color match. This method is based on converting the hemoglobin to acid hematin and then usually matching its color against a solid glass standard. The test characteristics includes the health clinics, the mobile laboratories, the field-based surveys with the sensitivity of 85% and specificity of 85% each. The equipment and the supplies required is Sali's hemoglobinometer, the blood pipette and the hydrochloric acid, small glass rod, dropper. The hydrochloric should be 0.1 molar hydrochloric acid and the distilled water. Now let's have a look at the test procedure of hemoglobin. Fill the Sali's graduated cylinder to the 2G mark with the dilute 0.1 m hydrochloric acid, approximately 0.15 ml. Now clean the fingertip with the cotton wool soaked with the 70% alcohol. Allow the alcohol to dry. Obtain a drop of blood by puncturing the fingertip with a sterile lancet. Now draw the blood to the 0.02 ml mark using the Sali's blood pipette. Add the blood to the dilute acid. Mix the blood and then acid thoroughly by flushing the pipette several times. Allow the acid blood mixture to stand for 5 minutes. Now hold the scale up to the light. If the color of the solution is same as in the light that of the colored standard given in the stand, the hemoglobin level is 4 gram per deciliter or less. If the color of the solution is darker than the colored standard, continue to add the distilled water drop by drop. Now, first mix the solution with the glass rod after each drop is added and compare the solution to the colored glass standard. Now keep adding the distilled water until the color of the solution matches with the color of the glass standard. Hold the scale up to the window which assess the color match. Now once the color matches, hold the instrument closer to eye level and record the value of the hemoglobin indicated on the side of the tube by the level of the fluid. The advantages of this method are that it is an inexpensive method and easy to perform. The electricity is not required to conduct this test. However, the disadvantages of this method is that the color matching is subjective. The color of the standard will fade with time and needs periodic calibration to determine the correction factor. The samples can only be measured one at a time, and after a prolonged use, the numbers on the graduated cylinder fade and are difficult to read. The Cyanmethemoglobin method is an internationally recommended method for determining the hemoglobin, and it requires dilution of the blood. The Cyanmethemoglobin method is the most accurate method of measuring hemoglobin, and it is considered the reference standard. A measured sample of whole blood is added to the Drapkin's diluting fluid. The red cells are hemolyzed and the hemoglobin is converted into a stable compound, hemagglutinoside. This sample is placed in a colorimeter, and the amount of light that is absorbed by the blood sample at a certain wavelength is proportional to the amount of hemoglobin in the blood. The test characteristics are: the appropriate setting is laboratory setting, its accuracy is sensitivity of about 100% and specificity of 100%. The equipments and the supplies include cuvettes, test tubes, pipettes calibrated to deliver 0.02 ml, pipettes calibrated to deliver 5 ml, photoelectric colorimeter, Drapkin's diluting fluid, standard solution of hemoglobin at 15.0 gram per deciliter of blood, graph paper and a watch. The sample is the venous blood collected in EDTA, and the test procedure is preparing a standard calibration curve. This procedure should be done frequently, every week or two, and when fresh reagents are prepared. Ensure that the colorimeter is used with a yellow or the green filter. If the spectrophotometer is used, adjust the wavelength to 540 nanometer. Switch on the colorimeter or spectrophotometer and allow it to warm for 10 minutes before the use. Prepare the Drapkin's diluting fluid. Label 5 tubes 1 through 5. Be sure the matched colorimeter test tubes or cuvettes are cleaned before using. Prepare the dilutions by adding the standard solution of hemoglobin at 15.0 gram per deciliter to the Drapkin's diluting fluid in the quantities indicated here. Next, mix the solution and allow them to stand for 5 minutes. Fill a clean cuvette with the solution from tube number 5, that is Drapkin's diluting fluid. Wipe the outside of the cuvette and remove any bubbles in the solution by tapping the cuvette. Place the cuvette in the colorimeter, ensuring the transparent sides are facing the light path. Adjust the colorimeter to 0 absorbance. Fill 4 additional cuvettes with solutions from tube 1 through 4. Place each cuvette in the test slot and read its absorbance from the colorimeter. Record the result. Plot the absorbance reading of the diluted standard against their known concentration of hemoglobin. From the graph values, prepare a table of hemoglobin value from 2.0 to 18.0 gram per deciliter with their corresponding absorbance values. The hemoglobin estimation can be done by the steps like label the tubes with subject identification numbers, use the pipette to add 5 ml of Drapkin's diluting fluid into each tube, draw the venous or capillary blood to the 0.02 ml mark of Sali's pipette. Place 0.02 ml of blood in 5 ml of the Drapkin's or the modified Drapkin's diluting fluid. Flush the pipette several times in the diluting fluid to ensure delivery of the entire quantity of the blood. Inspect the blood and dilute the fluid mixture for cloudiness prior to taking the measurement. Cloudiness can be minimized by addition of a small amount of detergent to the solution. In next step, mix the content of the tube thoroughly and let it rest for 5 minutes at room temperature so that reaction will be complete. Fill each cuvette with the amount specified. Use Drapkins or modified Drapkins diluting fluid to zero the spectrophotometer at 540 nm or for a control match in a colorimeter. Read and record the absorbance of each specimen. Now let's have a look at the advantages of this method. All forms of hemoglobin except the sulfur hemoglobin are converted to Cyanmethemoglobin. The visual error is not there because no color matching is required. Cyanmethemoglobin solution is stable and its color does not fade away with time, so readings may not be taken immediately. A reliable and stable reference standard is available from World Health Organization for the direct comparison. Some of the disadvantages of this method are that the diluted blood has to stand for a period of time to ensure the complete conversion of the hemoglobin. The potassium cyanide is a poisonous substance, and that is why the Drapkins solution must never be pipetted by the mouth. The abnormal plasma proteins cause the turbidity when blood is diluted with the Drapkins solution. A high leukocyte count also causes the turbidity on dilution of the blood. Portable hemoglobinometer. This method requires lysis of the blood. The portable HemoQ hemoglobinometer is most commonly used device for quantifying the hemoglobin in the field surveys. The HemoQ is an example of a rugged, portable and accurate hemoglobinometer readily available for use. The HemoQ uses a disposable cuvette that is treated with chemicals that ruptures the red blood cells wall and combine with the hemoglobin to form a compound that can be measured photometrically. The test characteristics include the appropriate setting include rural setting for the surveys and where high accuracy is important. Its sensitivity is 85% in field settings and 100% in the laboratory setting. Its specificity is 94%. The equipments and the supplies required for this test are HemoQ instrument, disposable cuvette, nickel-cadmium batteries, standard control cuvettes. Now let's have a look at the test procedure. Move the switch at the back of the HemoQ instrument to the power on position. Pull out the cuvette holder to insertion position. This is noted by a distinct stop which should not be exceeded. Calibrate the hemoglobinometer using the standard cuvette provided. Fill the disposable cuvette with the blood drop by placing the capillary tip of the cuvette on the blood drop. Place the filled cuvette in the cuvette holder and push it into the device until it stops. After 45 seconds, the hemoglobin value is shown on the display. There are some disadvantages and advantages of this method. The advantages include that it's a portable, lightweight and a sturdy instrument. It's accurate, the rapid results that requires no calculations. It is simple to use and it minimizes exposure to the blood. On the other hand, the disadvantages include the high cost of the instrument and disposable cuvettes. The disposable cuvettes cannot be reused. In addition, the high humidity can adversely affect the performance. The hemoglobin color scale method, that is WHO color scale. It works on the principle that it does not require lysis or the dilution of the blood. This method is developed by the World Health Organization in 1995 based on the principle that the color of the drop of blood could reliably indicate the closest range of the hemoglobin content. The color would be matched against the predetermined shades of the red. The test characteristics are: the appropriate setting include most useful for screening in the rural setting with the accuracy of sensitivity 60% at 10 gram per deciliter and the specificity of 60% at 10 gram per deciliter. The equipment and supplies required for the test involves the filter or blotting paper that allows the absorption and rapid drying of the blood drop, a standardized color comparison chart that represents ranges of hemoglobin level, some sterile lancets, 70% alcohol and the cotton wool. The test procedure include the step 1, collect the blood drop onto the filter paper and wait a few seconds until the blood spot has almost dried. When the blood spot is no longer shiny, hold the filter paper behind the standardized chart so that the blood spot is visible through the hole in the chart. After the blood is dropped onto the filter paper, the reading must be performed within 10 to 15 minutes because the color of the blood spot on the filter paper will continue to darken as it dries. Find the color on the chart that most closely matches the color of the blood on the filter paper and record the result. The advantages of these methods are the inexpensive technique, rapid and a simple technique. There is no need of reagents, it's quite portable, the electricity is not required, the filter paper and color chart are durable if properly maintained and stored. However, some of the disadvantages of this method are that the chart is supplied with a limited quantity of the filter paper. Other types of paper cannot be substituted because they are not calibrated with the chart. The scale may become contaminated with blood over time. Lighting conditions influence the interpretation of the result. And the last is that the size and thickness of the blood spot, temperature and humidity all affect the drying time, which in turn affects the color. The next is copper sulfate method. This method does not require lysis or dilution of blood. The copper sulfate test is based on the fall or the flotation of the whole blood when dropped into a copper sulfate solution of a known specific gravity. The drop of the blood will either float or sink depending on whether it's lighter or heavier than the copper sulfate solution. The appropriate setting for this test is the screening of the blood at screening programs such as for the blood donors or the antenatal clinics. Its sensitivity is 87.5% and specificity is 99%. The required equipments are analytical grade copper sulfate, weighing scale, amber tinted cylindrical bottle, graduated transfer pipette, volumetric flask, clean distilled water, EDTA capillary collection tube, sterile lancet or needle and the syringe. Test procedure: there is a need to prepare a stock solution with specific gravity of 1.1. Test procedure includes that there is a need to weigh the 170 gram of copper sulfate and add this quantity to a larger flask. Add 1 liter of distilled water to the copper sulfate in the larger flask. Rotate the flask to dissolve the copper sulfate. If the hydrometer is available, confirm that the specific gravity of the solution is 1.100. Store the stock solution in an amber bottle. Collect the blood in a capillary tube or EDTA treated capillary tube. Select the standard solution that will give the cutoff value needed to assess the level of hemoglobin of the subject. Release a small drop of whole blood from 1 cm above the standard solution. As soon as the blood enters the copper sulfate solution, observe the drop for 10 to 15 seconds. If the blood drop sinks immediately, its hemoglobin value is greater than that of the hemoglobin gram equivalent of the copper sulfate solution. If the blood drop floats for 10 to 15 seconds, the hemoglobin value is equal to or less than the hemoglobin gram equivalent of the copper sulfate solution. There are some advantages, including the inexpensive method. The interpretation is more objective than visual color matching methods. Several solutions can be prepared to measure a range of hemoglobin level, and there is no need of electricity. However, there are many disadvantages, like it is difficult to obtain an analytical grade copper sulfate in rural setting. It provides only range of hemoglobin levels. Several solutions need to be prepared and the error increases progressively with continued use of old solution. Some of the other methods for hemoglobin estimation are oxyhemoglobin method. Here in this method, hemoglobin is converted to the oxyhemoglobin by reaction with the ammonia and the color of the solution is measured in the photocolorimeter. The advantage of this method are that it is a simple, quick and its reliability is not affected by the increased bilirubin level. The another method is alkaline hematin method. The blood is converted to the alkaline hematin by addition of a alkali such as the sodium hydroxide and the color is measured in a colorimeter. It gives a true estimate of the hemoglobin and is not affected by the presence of plasma, protein and the lipids. Another method is the measurement of oxygen carrying capacity. The oxygen combining capacity of blood is 1.34 ml oxygen per gram of hemoglobin. This is a measure of the function of the hemoglobin. It is not used continuously as it is not very practical and gives result 2% lower than the other methods. The iron content of the hemoglobin is another method. This method gives us the hemoglobin estimations by measuring and converting the hemoglobin using the formula of 0.347 mg iron per 100 mg of hemoglobin. It is, however, impractical for the routine use. The data presentation. This section discusses briefly the data presentation of a series of hemoglobin estimation from population survey. The fundamental statistical concepts that are discussed in this section are mean, variance, standard deviation and coefficient of variation. These estimates are essential to understand the measure of dispersion in a sample data sets and gives an estimate of representativeness of the universe. Mean. The mean or average is a value which is typical or representative of a set of data. It takes account of all observations and is determined by calculating the sum of all the observations and dividing the number of observations. Variance is a measure of the dispersion of results around the mean. It is the average of the sum of squares, that is the average of the squared deviation, where the mean is subtracted from each value. Because it's a squared quantity, the value of the variance will not be assigned the same units of the measure as the individual observation. For example, gram per deciliter of the hemoglobin. Thus, its use is limited. The standard deviation. It is also a measure of dispersion about the mean and is the square root of the variance. The standard deviation is the most common measure of dispersion because it's expressed in the same unit as the original observation. It's determined by taking the positive square root of the variance. And last is the coefficient of variation, which is a measure of the relative precision that is determined by dividing the standard deviation by the mean and is expressed as a percentage. The coefficient of variation is normally used for determining the precision of several measurements of the same sample. Now let's summarize the module. We have studied so far that hemoglobin is the major constituent of the red cell cytoplasm, accounting for approximately 90% of the dry weight of the mature cell. It's comprised of the heme and globin. Hemoglobin is important substance in the human blood cells, the deficiency of which may lead to the anemia. The anemic conditions in the women and children across the world is a serious concern. This helps in identifying the level in preliminary and important investigations. There are several methods available for the hemoglobin estimation. In anthropological fieldwork, the estimation of hemoglobin level has been entrusted with the methods which involve the least logistical issues and have reasonable sensitivity and specificity. In the given module, we have discussed so far the 5 methods of hemoglobin estimation. That is the Sali's acid hematin method. This test is the most commonly used and trusted method for the hemoglobin estimation in resource poor settings and the field situation. The distilled water is added to the graduated cylinder in this method containing a blood sample until the color of the diluted blood sample matches that of the glass standard. We keep diluting the solution. The quantity of the dilute acid added to the determined by the hemoglobin level of the blood sample. The Cyanmethemoglobin method, this is the internationally recommended method for determining the hemoglobin, and it requires the dilution of the blood. The Cyanmethemoglobin for the hemoglobinocyanide method is the most accurate method of measuring the hemoglobin and is considered the reference standard. The portable hemoglobinometer is an example of the rugged, portable and accurate hemoglobinometer readily available for the use. The hemoglobin color scale method, WHO color scale method, this method does not require the lysis or the dilution of the blood. This method is developed by the World Health Organization in 1995 based on the principle that the color of the drop of blood could reliably indicate the closest range of the hemoglobin content. The color would be matched against the predetermined shades of the red. The copper sulfate solution method, this method does not require the lysis or the dilution of the blood. The specific gravity of the blood is influenced by the red blood cell volume. The copper sulfate test is based on the fall of the whole blood when dropped into a copper sulfate solution of a known specific gravity. The standard copper sulfate solutions are used to determine a particular hemoglobin level. Thank you.